Hello students, welcome to EPG Pathshala. I am Dr. Vel Pandian from All India Institute of Medical Sciences. Today I am going to talk to you about a module of Working Principles of HPLC and its Application, Part 2. This is from the paper Techniques in Molecular Biophysics 1. In this module, what we are going to see is that sample preparation methods for HPLC and new method development, HPLC process optimization, method validation, and the regulatory requirements for developing a successful HPLC method. Well, sample preparation methods in HPLC analysis is a very interesting field, and it is a very complicated field. So all the more important is that whenever we are trying to develop a method for a sample preparation, there are several factors. The important factor is removing the interfering matrix. For example, you take blood. Blood is one of the dirtiest matrix you could think of. The blood is containing RBCs, it is containing plasma. Plasma is containing much of albumin. Now you see the complication is about you have so much of lipids, you have so much of protein, and you have so much of other interfering elements. So how to purify your analyte to some extent from this matrix, interfering matrix elements. So in this task we use different methods to some extent to remove the interfering matrix so that we can get a relatively good amount of good type of sample for getting further separation done in the HPLC. And you need to remove a particulate matter. This is very important. Otherwise, the particulate matter which is present in the sample, especially in environmental samples. If you take a water sample, for example, if you just try to sample water from a river, this is containing a lot of sand, dust, and all other purified particulate matter. It might be containing bacteria, it could be biological or it could be natural. These particulate matters will be interfering in the analysis. First of all, this go and choke the column. The back pressure of the column increases tremendously and it will cause HPLC to shut down, or it might even break the plunger of your pump. Therefore, removing this particulate matter is again another task. Sometimes the amount of substance which is present in the sample is very, very small. For example, you are trying to do an environmental analysis about remnant contaminants, and the contaminants, you are trying to remove the contaminants, but some amount of contaminant is left, the remnant is there. So what you try to do is you try to concentrate these solutes to decrease the volume, so that you are trying to multiply the concentration in that solution. And sometimes you need to, this is very important because HPLC, in which the detection, the type of detection method which is being employed is entirely dependent upon the property of the analyte which is of interest. Sometimes, for example, if a molecule is having good molar extension coefficient, you are lucky enough to get good peak in the ultraviolet spectroscopy. Otherwise, it doesn't have any conjugated double bonds, or it doesn't have any required molar extension coefficient, you will not get a good sensitivity. For 1 microgram you get a small, you may not get any peak at all. You may not get any peak. Or when you keep on increasing, you might get peak somewhere around 20 microgram or 40 microgram, which is of no use if your analyte concentration is 100 nanograms. Therefore, concentration step is employed here. It's completely, you have to work with a logic to make this possible. Sometimes the molecule of interest is present in lipids, and the whole analysis is very complicated in that case. You need to opt for some specific purification methods like solid phase extraction types, which we will see subsequently. So the criteria for good sample preparation is the molecule of interest should be in solution compatible with the mode of HPLC selected. It should be stable, it can be recovered quantitatively, and recovered in a minimal volume. So these are all the criteria for a good sample preparation for HPLC. So the options we conventionally we have for sample preparation are centrifugation, filtration or ultrafiltration, selective precipitation, liquid-liquid extraction, solid phase extraction, dilution or concentration, or some other when you try to go for some sort of ion exchange and immuno precipitation, etc. Centrifugation is popularly employed for removing the particulate matter. Filtration or ultrafiltration is employed to get rid of this solid matrix, or sometime even we will even try to remove some. For example, you try to work with reactors, bioreactors, in which you are having the inoculum. From the inoculum, you wanted to get the ultrafiltrate to do the analysis. Here you use the ultrafiltration techniques. We have small ultrafilters having a different molecular weight cutoffs, which are used to selectively remove the subject, which is a molecule of your interest. Or sometimes we go for selective precipitation. For example, you take plasma. You try to remove the albumin as a major protein contaminant by using different methods. For example, somebody might like to decrease the pH. pH causes precipitation of proteins. Sometimes they use sometimes we use heavy metals for the precipitation. Sometimes we use more than 70% of organic compounds to precipitate the protein. The solubility goes down at precipitate. Sometimes we use high concentration of salts to precipitate. We go for liquid-liquid extraction, and sometimes, so for example, your substance is in a liquid form in the plasma, and you use chloroform or methyl chloride, try to do a liquid-liquid extraction in which you try to take out selectively your compound by using an organic liquid, and you evaporate the organic liquid to fetch the compound of interest, which can be subsequently dissolved in the mobile phase, which can be used for an HPLC. Next is solid phase extraction, which I will show you in the subsequent slide. How do we do solid phase extraction? Sometimes we go for dilution. These are very highly concentrated substances. Sometimes pharmaceutical formulations are highly concentrated. So in that case, we have to go for dilution, or environmental samples are highly diluted, we go for concentration. Sometimes we use specific columns for ion exchange columns, or we use immunoreactive columns to capture some sort of a specific bioactive compounds. I'm going to explain you how sample preparation is done with the help of solid phase extraction. I have already explained to you about liquid-liquid extraction, about precipitation, selective precipitation, and other methods and all. But today, much of separations are reached by using this solid phase extraction. Is one of the widely used preparation method in today's chromatography science. Here, the solid phase extraction cartridges are used, where you have again, it's like a mini chromatography. You have syringes, or 2 ml syringes or 5 ml syringes filled with silica, especially C18. Silica is filled up with having a mesh size of around 20 micron particle size, 40 micron particle size, is packed of 1 gram, 100 milligram or 200 milligram, depends upon your separation, along with a sintered disc from on top, in the bottom and the top. Where the sample is loaded, your sample is a biological sample. This is used for semi-purification of your sample. You load the sample and you elute it with a particular solvent, so that selectively the hydrophobic compounds get absorbed on the silica, on the C18 matrix. Then all the water soluble compounds goes out. Then selectively, if you put methanol or acetonitrile, you will be able to leach this compound which are stuck to the column bed selectively. So first the method goes is that you do a conditioning of this SPE, that is solid phase extraction cartridges. Usually this is done under suction, so there are assemblies available to do this extraction possible. And conditioning the column is conditioned with acetonitrile, and so that whether what you are trying to look for a partition type or it is an adsorption type, based upon which condition changes, then the biosample is slowly charged on it. Then You add a simple aqueous solution like water or buffers are used to wash the biosample, which usually the drug is or a compound of your interest is bound to plasma. Suppose you are trying to elute plasma, there's albumin which is having the bound compounds which is getting transferred to this C18, and when you're washing, this albumin goes off. There's the interfering matrix. Now the albumin goes off. After that you apply suction to dry the column. Now you chain the tube and you add only pure methanol which elutes the bound molecules of your interest into the tube. Thereby you get a semi-purified sample which can be subsequently used for your analytical separations. Ultrafiltration assembly. Ultrafiltration is another way of purifying the sample or making it ready for HPLC. This is a small microcentrifuge device. It's in the same microcentrifuge tubes, which will be having a small container as shown in the figure, a small tumbler-like figure, in which you load the sample and you put it inside the MCTs, that is the microcentrifuge tubes, and subject it for centrifugation. So here the ultrafiltrate comes out in the bottom because you apply centrifugal force. So due to this centrifugal force, the aqueous phase is getting separated and comes out. By using this kind of devices, you will be able to purify peptides, smaller molecular weight peptides from high molecular weight peptides, or you'll be able to get the ultrafiltrates. If you are interested in ultrafiltrates for some compounds, you'll be able to do that. Basically, you try to remove high molecular weight protein, and here you have a variety of cutoffs, which could be 20 kD cutoff or 10 kD cutoff or 5 kD cutoff. You use it according to your method, your protocol, or your SOP, so that you'll be able to get a compound which is of interest. So this is how the ultrafiltration devices works for purification of compounds in HPLC, a semi-purification of compounds in HPLC. As of now, we have seen about how do we prepare a sample for HPLC. Now we are going to see how we are going to develop a new method in HPLC of a known compound, new method for a new compound, or how we develop a method for it. And the relevant information, what I would look forward for any new compound or a known compound, is that I look for it for its molecular weight. If it is a proteinous compound having a higher molecular weight, I may try to choose appropriate column for that. But if it is a smaller molecule, which is less than 500 molecular weight or less than 1000 molecular weight, in which I may like to choose the C18, but I would like to look for it for its solubility, that is chemical nature. Whether it is soluble in water or soluble in methanol or soluble in hydrophobic solvents like chloroform or methyl chloride. If it is soluble in methanol, it suits me well. I would choose C18. If it solubilizes only in water, and it is highly soluble in water, then I may not like to choose C18 or C8. I may like to choose Hilly column, that is hydrophilic interaction chromatography. Or if it is R, I will look forward for its chemical nature much more to know about pKa of this compound or partition coefficient of this compound. Partition coefficient is as a ratio, a mathematical ratio, exists between a compound when it is trying to solubilize between two immiscible liquids. Usually it is octanol water partition coefficient. With knowing this value, which will help me to choose a compound or column which is of my interest to separate this compound. Next thing is I may like to understand about its UV absorption characteristics or fluorescent characteristics, about the particulars of this by using spectrophotometer or a fluorescent spectrophlorimeter. Or if it is not having any one of this property, I may like to look forward for a Dux potential or look forward for the refractive index to use appropriate detectors. So this relevant information is very much essential before you touch HPLC to go for method development. When I look forward for a new method development, what I would do is, okay, now my compound is very much non-ionic. It doesn't have any acidic or alkaline groups, is pretty neutral. Like for example, let me just take acetone with benzene or acetone with toluene, or if I just look forward for an acidic compound like let me take a drug like ibuprofen, which is an acidic drug. So I may try to choose, having a good solubility in methanol, which ensures me that I can use octadisylsilane, that is C18 column. What I will try to do is, I will try to understand by injecting a small amount or 10 microliter of acetone. I will try to understand when the peak is coming out. This acetone is not retained in the C18 column. Therefore, wherever it is emerging out, I would consider as the void volume, that is a VO. Now, I will dial 210 nanometers or 254 nanometer, depends upon the compound which I am going to analyze. Then inject the sample in pure methanol. I will try to run that 1:1 acetonitrile methanol. I will inject 10 microgram of sample and observe when it is coming out. If at all it is coming out or not, I will try to understand 1:1. Now, after that, what I will do for method development is what is called a scouting. So I will try to run run the same sample of 100 microgram in acetonitrile, 100% acetonitrile, followed by next run I will give with 80% acetonitrile, 20% water, and 60% acetonitrile, 40% acetonitrile, and 20% acetonitrile with water. So by this way, I will come to know slowly when am I getting the peak away from V0. And sometimes if I inject 20%, I may not get the peak at all. It must be stored in the column. When I keep on increasing acetonitrile, this will be coming out. Therefore, it is very important you start from 100% acetonitrile to go towards lower concentrations. Otherwise, you will be searching for peak. Where it will be retained in the column, you won't see the peak coming out. By this way, before I am doing this, I know very well about what is a character, what detector I am supposed to use. That is very important. Otherwise, if the compound doesn't have any absorbance, you won't be able to find out in the chromatographic run. If RT reaches beyond void volume, I would start optimizing it. I would try to tune with the pH to make the HPLC method done. If the compound is still coming near void volume in C18, then I would go by changing the column to C8. If run is good, separated peak looks for the following: a peak shape, a peak base, and peak sharpness. All three characters I will look into it. If the peak is tailing, or if it's having a fronting, or it is having dog or rabbit ears, or fronting, tailing, bell-shaped curve, all these things comes up, then I will start working with the chemistry. Now I try to go with a pKa. Either I go little up or little down from using the pH. I look forward for a beautiful peak which is coming out of HPLC. Typically, the buffers used in HPLC are phosphate buffers. To some extent, acidic pH, you use 20 millimolar phosphate buffers are much more preferred. Otherwise, sometimes the phosphate buffers would be will be crystallized when you just mix it with acetonitrile or with organic solvents. One has to be very careful, and HPLC should never be left in a buffer. Otherwise, the buffers will be corroding the plunger seal, as well as the piston will be getting eroded, and it might cause the HPLC jam. The diammonium hydrogen phosphate is used, sometimes citrate buffers, acetate buffers, but citrate buffer and acetate buffers are having one problem: they have high UV absorbance. Therefore, preferably potassium phosphate buffers to an extent of 20 millimolar, adjusted to the suitable pHs, can be used for HPLC separations. If a compound is very aliphatic in nature, acidic, and it's not able to elute properly, I may look for a strong hydrophobic ions to make ion pairing with the compound. Such compounds are tetrabutyl ammonium hydrogen sulfate, 1-octane sulfonic acid, pentane sulfonic acid, heptane sulfonic acid, used from 20 to 50 millimolar to make that adduct. Along with that, to enhance the separation in C18 column. As of now, we have seen about sample preparation. We have seen about how to handle, developing method in HPLC, etc. Now, after developing methods, now we are going for HPLC method validation. And this validation, there are several parameters are there in HPLC which explains how the method should be validated. Let us see one by one. As far as method validation is concerned, there are certain regulatory guidelines explains about what it's supposed to be. The guidance of interpretation of in 45,000 series of standards of ISO IEC Guide 25, International Conference of Harmonization ICA-GCP guidelines explaining technical requirements for the registration of pharmaceutical for human use, or United States Environmental Protection Agency explanation for validation, or US FDA validation, or USP validation, and any of these validation protocols talk about method validation for HPLC. When we follow these protocols, it's very good for us to get the reproducibility and acceptability of the method for any of the purpose for which it is intended to. The strategy for method validation is first to develop a validation protocol or operating procedure, define the application, purpose and scope of the method, define the performance parameters and acceptance criteria, defining validation experiments, verification of relevant performance characteristics of the equipment, qualify materials, especially standards and reagents about their standards, their qualities especially, and perform pre-validation experiments. This is a strategy for method validation. It has to be done one by one. While deciding the strategy for the method validation, adjust the method parameters and acceptance criteria if required, because you have to work within that parameter, or you have to optimize the method to reach that criteria. Perform full validation of experiments. Develop standard operating procedures for executing the method in routine. Suppose if you are planning to do subsequent analysis. For as a matter of fact, whenever we try to do the experiment, it is always important we need to run the standard calibration curve to see is this within the limit or not. It should not be repeated, it should not be done that one day I have done a standard calibration curve and second time when I do the experiment I am not going to do that. It's not a way how the SOP is to be made. SOP is to be made and it should be followed every time whenever we try to run that method. And to define the criteria of revalidation in case if it is required, define the type and frequency of system suitability test and analytical quality control checks for the routine. So in between the run, always give a quality control sample to ensure that how much this method is keep on maintaining, how long this method is maintaining the same linearity of the analytical calibration curve which has been plotted and which has to be used for the test what you are trying to do. And document validation, experiments, the results and the validation report. These are all the strategies for method validation. Sometimes revalidation is required for HPLC method in case there is any change in synthesis of drug substance or any synthesis molecule, the molecule which is getting synthesized. Because when you're trying to deal with a molecule, always HPLC separation happens based upon the separation factor, that is between how good it is capable of resolving between two peaks of closely related substance. Here, if you are changing the process of synthesis, again it has to be revalidated. And the changes in the composition of finished product. Sometimes the chromatography is done for the finished product and for which the composition is changing. Therefore, interfering substances changes. Changes in analytical procedure. Any change in analytical procedure calls for revalidation. Sample matrix and instrument type changes. Suppose today I am working with one HPLC. Tomorrow I am going to another lab to do another HPLC. I need to revalidate my method. If system suitability tests or results of quality control sample analysis are out of a preset acceptance criteria, I am keep on working with an HPLC. I am not getting the parameters not within the limits, indicating that there is something going wrong. For example, aging ultraviolet lamp, deuterium lamp is having a limited lifetime. In continuously, when you are working on sample, 6 months before I have done an experiment, extensively the HPLC is on operation. 6 months later when I go and do it, if aging deuterium lamp causes decreased energy continuum, it can cause drop in the parameters which calls for revalidation of the method. The parameters for the method validation. I have shown you several international agencies having their own criteria for method validation. It is usually defined in different working groups of national international committees. Unfortunately, some of the definitions are different between different organizations. Therefore, an attempt for harmonization was made to pharmaceutical application through International Conference of Harmonization, that is ICH GCP guidelines. Before we go for complete validation, first thing what we need to look for is system suitability. System suitability tests are an integral part of chromatographic method development. Now, these are all the factors which I have, like some of them I have already explained to you, but these tests are very important to understand. I mean, the method what you have developed is fit to carry forward for the method validation, because method validation, by the time of method validation, you must have already spent a lot of time in doing that work. Finally, if they are not fitting well, you have to completely redo it again. Therefore, it is important you do system suitability test before you go for complete method validation in HPLC. So, some of these tests include in system suitability all capacity factor calculation, repeatability, peak resolution, tailing factor, and theoretical plates. These parameters will explain about its system suitability. Let us see one by one. The capacity factor, as I have already explained you, what is void volume. Now my job is much easy. The capacity factor, I know usually it is divided by this formula, capacity factor K is equal to V1 by V0 by G0. This V0 is a void volume. V1 is a retention time in which your peak is coming up. When you calculate it, it should be 128. It should not be less than this number, which shows, well, it is capable of in resolving well away from V0. The next comes is tailing factor. If a peak shape is good, then the tailing factor should be less than 2. This is calculated by 2a by a plus b from the peak. This is what I have this is shown in the figure, and it is calculated, by the way, it should be less than 2, which shows that the tailing factor is less. You should not be taking account of a bell-shaped curve which is coming up having a very prolonged tailing factor for your analytical separation or to be taken for further validation purpose. Next parameter is height equivalent to theoretical plates. This height equivalent to theoretical plate is calculated. It should be good in number, which should be more than 2000 for analytical separation. If it is as much as possible in number, you get a better separation. So usually people prefer to go for 5 sigma, and 5 sigma, it is 4.4% of its height. You try to measure the width of the peak, and using the formula 25 into retention time divided by width at 5 sigma square will give you the number of plates it is capable of. If it is more, you get more. Otherwise, again, you need to work upon in the chemistry to improve the plates so that you get a better analytical separation. Then comes selectivity. Between two peaks, how good it is capable of. So capacity factor of first peak, capacity factor of second peak, you need to calculate. So K2 is the capacity factor of second peak, and the K1 is the capacity factor of peak 1, which V2 minus VO by V1 minus VO, which gives you a difference more than 1. That shows that they are well separated. If the number increases more, you get much better separation. If the second peak is coming at is far away from the first peak, you get very good selectivity in separation. And the separation factor is calculated by using this equation. Separation alpha is equal to this equation, and it should be between 1.1 to 2 to show the separation is good. Next comes is about repeatability. Repeatability is that when you try to repeat the experiments, it should the relative standard deviation should be less than 1%, right? And for which the number of samples should be more than 3 to 5 is desirable over the period. So over the concentration range. So the repeatability is another important parameter that batch to batch variation. Sometimes when you go for injection of manual injection, the errors of relative standard deviation goes up, and it should be minimized as less as possible so that you don't get much of problem in the injection and your hands are set for that. In autosampler, it's much easier. when you are continuously injecting same concentration on the same volume over the period over about more than 5 injections, it should give the same area and with less than 1% of deviation. Next is resolution. Resolution is between, it should be more than 2, between 2 the peak of interest and the closest eluting potential interference peak. For example, it could be internal standard, or it could be a metabolite or a degraded compound which is coming next to it, which is calculated by this equation. To simplify, United States Pharmacopoeia shows about 8 steps of method validation, which includes accuracy, precision, specificity, limit of detection, which is also called as LOD, limit of quantification, that is LOQ, linearity and range, ruggedness and robustness. If you just try to follow these 8 steps, you will be able to make comfortably a good method for your specific compound in HPLC. I have already explained you about the parameters which are governing about method validation. Let us see one by one. First is accuracy. Accuracy, it is a measure of exactness of an analytical method. So it is explained by percentage recovered by assay by spiking the samples in blind matrix over 3 different levels. So the percentage recovered by assay, for example, if we are going to spike a sample which is matrix, which is about 10 microgram, but in the assay what you are getting is about 90 microgram. We are getting it is out of 10 microgram you are getting 9 microgram. That shows your method is capable of recovering about 90%. Is pretty good. So this is what the accuracy is about, spiking samples in blind samples over 3 different levels. So the 3 different levels are usually low, intermediate and high. Next comes precision. Precision is, we define that it is a measure of repeatability of an analytical method under normal operation and is normally expressed as related standard deviation for a statistically significant number of samples. Usually n is equal to 3 is used over the ranges of low, intermediate, high, or sometimes even use 5 level calibration today. Nowadays we use it. So now what you do is that it has to be repeated intraday, within the day and within the days. So intraday and intraday variation is analyzed by injecting 3 times of n is equal to 3 and trying to calculate the RST between them, and it should be within the limited criteria of ICH GCP guidelines. Next comes to specificity. It is the ability to measure accurately and specifically the analyte of interest in the presence of other components that may be expected to be present in the sample matrix. Here the sample matrix is the sample matrix is a pretty complicated matrix where it's how accurately your method is capable of measuring in a sample matrix. If it is a blood or it could be plasma, and this decides specificity is decided by how good is your analytical method. The diode detectors comply with this issue because the matrix might be having several things. For example, blood is having cholesterol, it has bilirubin, it has several other compounds which are soluble, which are capable of soluble in chloroform. Suppose if we go for liquid extraction, and this will be showing. Although your analytical wavelength may not be able to see, at a different wavelength you will be able to see that. That is why diode detectors are capable of detecting the specificity of your method. Next comes limit of detection. This is often used by analysts, LOD and LOQ. Sometimes they say LLOQ or LLOD. So this lowest concentration of analyte in a sample that can be detected, not quantified or quantitated. That means your method is capable of detecting something. Usually this is in the rule of thumb, it is 3:1 ratio of signal to noise ratio. So basically you will be getting a signal as a noise without any peak, and first if the peak is getting elevated 3 times of a noise at a particular retention time where you are expecting your analytical peak is called as limit of detection. It can also be done by another equation. LOD is equal to 3.3 by SD by S, that is S is equal to slope, and SD is equal to standard deviation of a standard calibration curve. Next comes limit of quantitation. This is a very important factor which is going to determine the accuracy of your method. The lowest concentration of an analyte in a sample that can be determined with an acceptable precision and accuracy under operational conditions of the method. According to ICH, this is LOQ is equal to 10 into SD by S. As here you will be able to use 5:1 as a signal to noise ratio for limit of quantification, and using this you'll be able to decide up to what concentration your method is capable of detecting the analyte. Next comes linearity and range. So you always plot the standard calibration curve to see the linearity, how linear the curve between the range, a particular range, the straight line you are getting it with the concentration of the analyte with the response. Ability of the method to elicit test results that are directly proportional to the analyte concentration within the given range. The allowable range is 80 to 120% of the target concentration. So the correlation coefficient, what is expected is 0.994 to 0.999 is the correlation which is expected within this range. When we talk about ruggedness is something, a degree of reproducibility of the results obtained under a variety of conditions expressed as percentage RST. So that means if I am able to run the machine, I should be able to get a data, but my friend is running the same method in the same machine, he should also get the same data. Therefore, this is with the varying conditions, the method is repeated, different labs, at different analytical instruments and different temperature, etc. And the method should be capable of giving that much of ruggedness, and as a percentage RST it is expressed, and it should comply the criteria of ICH-GCP. Robustness is a capability of the method to remain unaffected by small deliberate variation in the method parameters, slightly changing percentage of mobile phase, or pH change, or ionic strength, or temperature. Suppose if I am setting the temperature at 20 degrees centigrade, I am setting this method at 25 degrees centigrade, it should work the same, it should remain the same. Or slight pH change, suppose if I am running with 3.1 or 3.2, it should not change. Ionic strength, suppose instead of 10 millimolar, if I am using 12 millimolar, method should not change. Percentage of mobile phase, especially those who are running isocratic run and using filtration assembly, when you are filtering the degassing the isocratic mobile phase on the next day, you will see a drop in organic concentration, and by that way method should not show much. That's what is expected from robustness. So all these parameters need to be optimized when you are running an HPLC. So in the overall exercise, we have seen about system suitability parameters and method validation parameters to help you in the HPLC analysis. And these are all the parameters which are governing separations in HPLC. So students, let me summarize what we have seen, what we have learned from today's module. I've explained to you about sample preparation methods, how to find a suitable method for you for developing your own HPLC method, and how to develop a HPLC method for that particular analyte, and if you develop a method, how to optimize the method, and after optimization, that means the peak optimization, we have seen about the method validation as per regulatory guidelines. Together, this will help you in successfully developing a method and validating it which suits your application. Thank you.